We only heard one person join. Maybe we're going to be a real small group today.
Quite possibly, that's how it seems.
Uh, Anyways, I take it you can hear me, so that's good.
Yes, I can.
Great. Oh, boy, that's really slow. My computer is going to be very slow because it's making the Zoom file from the lecture. Let's go up there. And, let me share my screen here. Oops, that's the lecture. Where the heck is the Zoom for? I hope that doesn't close the meeting. Nope. I'll close the meeting. Let's cancel that. There it is. All right. I had trouble finding the Zoom file because there's three Zoom files open and um, I was having trouble finding which one was the right one. All right, let me share my screen now. Yep, there's one of the Zoom files I don't want. I don't think I can get rid of that. I could get rid of that. And there we are in the lecture. We're only 83% done with that file. Let's go to the syllabus. There we go. All right, today is February 20th, and we are supposed to be working on Lab 9. Are there any questions before I begin? All right, let me remind you, your infectious disease project or your infectious disease paper is due at 11:59 p.m. this Saturday, and you must get it approved by me. There's a few people who haven't gotten it approved. If you don't get it approved by me, you'll get a penalty. So get it approved before you turn it in. And then we're going to talk about Lab 9, identification of gram-negative organisms. You should also be working on your unknown project. Any questions about what we're doing?
Just a quick question. Remind me, please, when the lab report for the Unknowns project is due.
The lab report for the unknown project is due on March 6th, right there. No,
Thank you so
sorry, it's
much.
March 8th, Saturday, March 8th. The unknown notes pages will close on March 7th. So you must have your unknown notes pages due or finished by March 7th. Because after that, they'll close. You may ask for a test on March 7th, but if you ask for a test, it must be a test where you can get the results within 1 day or less. Most of the tests require 2 days of incubation. One test requires 5 days of incubation, and one test 7 days of incubation. But any test that is 2 days incubation, you will not get the results in time. So if you ask for a test on the 7th, you must ask for a test where you get the results within 1 day or less. All right, good. The lecture page is now finished, and so my computer will speed up. I think I'm going to close that down because I don't need it any further. All right, let's talk about Lab 9, identification of gram-negative bacilli. Lab 9 does have some reading in Tutora, so please read page 131 to 132, on biochemical tests and bacterial identification, and then about 4 pages in Chapter 10, methods of classifying and identifying microorganisms. We do have some supplemental material today where we have some video clips for you to watch. And then the primary learning objectives for this lab are one, interpret the results for the following test. I don't know what's going on there. No, the IMVIC reactions. IMVIC is an acronym standing for indole I, methyl red, M, Voges Perscure, V, the I is just a letter in there, and then the C for citrate. All of these tests we're going to talk about today, the indole test, the methyl red test, the VP test, and the citrate. And I said VP because I may be butchering Voges Perscure. That's a Russian name, and I'm not Russian, so I don't know if I'm saying that correctly. And then we'll talk about the carbohydrate fermentation test, the motility test, and the hydrogen sulfide production test. We'll talk about all these tests today, as well as talking about the triple sugar iron slant test. This is a test where many things are going on in the test. And then the SIMS test, which is a deep, and the SIMS test stands for sulfide production, indole production, and then motility test. So SIM, sulfur, indole, motility. And then we'll look at the growth of bacteria on McConkey's plate. That's a test looking at two different things. And then the hectone and enteric agar plates. This is a test looking at three different things going on in the hectone and enteric agar plates. And then lastly, we'll use biochemical analysis to solve case studies involving gram-negative bacterial infections. In the last lab I graded, there were a number of errors in the case studies. You do need to know how to do case studies. There will be questions on the final on that, as well as I think the next quiz. So you do need to practice on case studies. So we're going to look at the family Enterobacteriaceae today. The members of this family are all small gram-negative bacteria, non-spore forming bacilli. They are all facultative anaerobes. They also all digest or ferment the sugar glucose, and they're all oxidase negative. There are 53 genera in this family, so it's a very large family. 24 of these genera have been known to cause human infections. Below are some of the genera in the family Enterobacteriaceae, which cause human infections. Some of them are very familiar to you, like Salmonella, Shigella, Escherichia. Most of these genera are normal microbiota of the intestinal tract, so we call them coliforms because you can find them in the colon. They are all opportunistic pathogens in that they cause urinary tract infections, wound infections, pneumonia, and septicemia. Common genera causing opportunistic infections include Escherichia, which E. coli is a member of, Enterobacter, Klebsiella, Citrobacter, and Serratia. So several of these are here, and these are genera causing opportunistic infections in patients. Several of the genera are associated with gastroenteritis, so these are not opportunistic infection or pathogens. They are frank human pathogens. These include organisms in the genera Salmonella, Shigella, and then E. coli, Serovar O157H7. This Escherichia coli has the antigens O157H7. This is a human frank human pathogen. If you get E. coli 157H7, you're likely to get gastroenteritis. Normal E. coli is found in the human intestines, and if you get it, you normally would not get sick. You'd have to have a very large number of normal E. coli to give you any sickness. And even large numbers of normal E. coli do not usually even cause sickness. But you have small numbers, like around 500 of 0157H7, you likely will get gastroenteritis. That's diarrhea and maybe um, throwing up. Salmonella and Shigella are two examples. Salmonella, for example, they are coliforms, but they are not found in the human digestive tract. But salmonella is very common in birds like chickens, and that's why you can find salmonella on undercooked chicken because it's in their intestines, and in the process of cleaning the birds, it may move from the intestine to the chicken skin, and then if you undercook the chicken, you can get salmonella. Any questions about any of that? All right, let's also talk about the family Pseudomonaceae. Pseudomonaceae includes Pseudomonas aeruginosa. All of these are similar to the family Enterobacteriaceae, and they are gram-negative short bacilli. However, the Pseudomonaceae are all non-glucose fermenting. They're all oxidase positive, and they're all aerobes. If you remember, the family Enterobacteriaceae are all facultative anaerobes. They're all oxidase negative, and they're all glucose fermenting. So all three of those can distinguish between the two families, Enterobacteriaceae and family Pseudomonaceae. Usually we do the oxidase test to separate out the Pseudomonas from the Enterobacteriaceae, but you could use any of the tests. I think the oxidase test is just quicker and easier to run than the glucose fermenting test, and also easier than the oxygen requiring test, meaning thioglycolate, row it in there, and then determine is it an aerobe or a facultative anaerobe. Pseudomonaceae is, at least aeruginosa, is an opportunistic pathogen causing opportunistic infections, particularly in immunosuppressed individuals, burn patients, and trauma patients. There's one good thing about Pseudomonas aeruginosa. It can cause disease in humans, but it is not very good at getting through the intact skin and the intact mucous membrane. But burn patients and trauma patients have their skin in particular, perhaps their mucous membranes also, damaged, and then Pseudomonas aeruginosa can get into them and then cause an opportunistic infection. Pseudomonas aeruginosa is also a healthcare acquired infection or a nosocomial infection, so we will talk about it in a later lesson when we're talking about healthcare acquired infections, also called a nosocomial infection. Any question about any of that so far? All right. It is important for microbiologists to be able to identify and perform antibiotic susceptibility testing on gram-negative bacilli that have been cultured from a clinical specimen. In this lab, we're going to talk about common biochemical tests that are used to identify gram-negative bacilli. And here are some of those tests that are used to identify gram-negative bacilli. We will not talk about all of them, but we will talk about some of them. The first test I'm going to talk about is the indole test. Some organisms produce the enzyme tryptophanase that can convert the amino acid tryptophan into the products indole and pyruvic acid. After overnight incubation, you then add Kovacs reagent to the tube, and Kovacs reagent will react with indole to produce a red color at the surface. E. coli tests positive for indole production. Klebsiella pneumoniae does not. So, in the case of tryptophan, which can be supplied just by the protein in the media, or you can add tryptophan to the media. And in the case the bacteria has the enzyme tryptophanase, you produce indole. Indole and Kovacs reagent make a red solution in which Kovacs reagent will be on top, the liquid. And I know that doesn't look very red, but this picture is a little bit off. I don't know, maybe it has a blue filter or something. Normally, Kovacs reagent is red in the solution above the liquid. And that's negative yellowish color, so that's correct. And that's a little purple. It should be more red. It usually is more red. All right, any questions about the indole test? Did I mention that? Yeah, I did. I said E. coli is positive for indole production. All right, let's talk about the MR test and the VP test. MR stands for methyl red, VP for Progres Voges Proscure test. Both of these tests are started by inoculating one tube with one organism. And then you incubate that tube, and then you split the tube into two, one half being for the MR test, and then one half being for the VP test. Both the MR and the VP test will look at the end products that result from the glycolysis. If you have acid products, the MR test will be positive. And if you have neutral products, the VP test can be positive. So both tests, the MR and the VP, are looking at the end products of glycolysis. As a general rule, if the MR is positive, the VP is negative, and vice versa. But occasionally, there are exceptions where you can have an organism which is positive in both the MR and the VP, or both negative in the MR and the VP test. All right, as I stated, the MRVP broth is one tube. You add one organism to it. You incubate it for 2 to 5 days. And then you split the tube into two. One tube will be running for the methyl red test, and the other test for the VP test, the other half of the tube for the VP test. MR test, you add methyl red to the media, and the media has a buffer in it. If the strong products are made, the strong acids are made for the MR test, then the acids will overcome the buffer, and the MR, when it's added, will remain red. If acids are not made in the tube, and then you add the methyl red will instantaneously convert to yellow, and then the tube would be negative, meaning the MR test would be negative. But if the tube remains red, then acid products are made from glycolysis, strong acids too, and that will be a positive MR test. All right, any questions about MR? And there we're looking at MR positive and then MR negative here. So MR positive, the tube turns red. MR negative, the tube stays or changes to yellow. Because methyl red is usually red when you add it. All right, the VP test, you take your half of the tube of the MR-VP test, and then you add Barrett's reagent A and Barrett's reagent B to the tube. You then shake the tube for about a minute to add oxygen, and then you let the tube sit for 30 to 60 minutes for the color to change. If the color changes to red on the top, then it is VP positive. If the color doesn't ever change and remains yellow, it is VP negative. And to be certain it's VP negative, you have to let the tube sit for 1 hour, 60 minutes. All right, any question about the MRVP 2 tests that start as one test? All right, let's move on. Citrate utilization test is looking at whether the bacteria can grow when citrate is the only carbon and energy source in the tube for the bacteria to utilize. The bacteria can utilize citrate, the bacteria will grow in the tube, and the tube will change color to blue. What happens is when citrate is used, basic products will be made, and that'll increase the pH, changing the color from green to blue. If the bacteria cannot utilize citrate, the bacteria will not grow on the tube, and the tube will remain green in color. So if citrate is used, the tube will change to blue color. If citrate is not used, the tube will remain green. Citrate, recall, is a ingredient of the Krebs cycle, an intermediate of the Krebs cycle. And if the bacteria can utilize citrate, it'll undergo the Krebs cycle and digest the citrate, making basic products, changing the tube to be blue. E. coli and Shigella are positive for the citrate test, meaning they can utilize citrate. And let me see if I can remember one which is not positive. Coccus luteus cannot grow on the citrate slant, and so it would be green. All right, any questions about the citrate test? If not, let's talk about the PR carbohydrate metabolism test. In the PR carbohydrate test, you're putting one sugar, or there's one sugar in the media, and then you put the bacteria in it. And if the bacteria can ferment that sugar, it will make acid products, which will change the color to yellow. And then you would score that as an A for acid. If, on the other hand, the bacteria can ferment the sugar, and in the process of fermentation, you also produce a gas bubble or gas production, as well as make an acid, you then score that tube as AG. So this tube can be negative, meaning no sugar fermentation, no gas production. It can be A, and that's acid production, or it can be AG, acid production and gas production by the fermentation of that sugar. In the carbohydrate fermentation test, you have different sugars. For our table, you could do glucose, or you could do lactose, or you could do sucrose, or you could do mannitol, or you could do indole. And I think those are all the sugars that you could do in your unknown project if you wanted to, because we have a table of it. But you could put another sugar in there, like beta galactose in there and see if the bacteria can ferment that sugar. Anyways, the carbohydrate fermentation test is the same. It's just that each test has a different sugar in it. And so you could call it the glucose fermentation test, or the sucrose fermentation test, or the mannitol fermentation test. But to make it simple, we're just going to lump them all together as the carbohydrate fermentation test. And in my unknown project, when I'm talking about the test, I don't call it the glucose test and the sucrose test. It's simply called the carbohydrate fermentation test. And it depends what sugar you have in there. And then you're only testing that one sugar. Any question about the carbohydrate fermentation test? All right, let's move on to the motility test. The motility test is a medium which has very low agar in it. And normally you have something like around 1% agar, and it's very stiff and thick. If you reduce the agar to something like 0.4%, and don't quote me on this, I don't remember exactly what it is. I think it's 0.4%, but it's some number much less than one. I know that. The agar then is semi-solid, and the bacteria can swim through it, even though it is a semi-solid. Jello would be an example of something that's semi-solid, where it can kind of wiggle. It does keep its shape, but it's not real good. Like if you were to pour it out of the container, it does keep the shape, but it kind of sags a little bit. Anyways, Jello would be another semi-solid media, and that's about what the motility medium is. It does use agar, but not very much, and so the bacteria can swim through it. If you stab inoculate the motility media and the bacteria only grow in the stab, you know that that is a non-motile species of bacteria or strain of bacteria. If, on the other hand, you stab inoculate the tube and then the bacteria grow everywhere because it swims everywhere, then the bacteria are very motile. And that's why that tube is cloudy everywhere. Let me blow this up. You can kind of see through, it's translucent, but you can kind of see through this region of the tube. You can't see through this region of the tube because it's cloudy. We also don't see the streak because it's hidden by the cloudiness of the media. And that's because the bacteria are very mobile. Now, you have a different result if the bacteria is slightly mobile. If the bacteria is slightly mobile, so this is our tube, we do have bacteria growing in the stab. So you do have that. But you also have, let's make the media across here. You also have bacteria growing outside of the stab. And it's usually a flare-like reaction, different sizes. We'll make a real big one here. And then a wide one here. And they don't have to flare the same. I gotta make this small here. All right, this is called flaring of the bacteria, where it's more than growing on the stab, but it's not growing everywhere. And that's because the bacteria are slightly motile. they will swim a little ways out, and that's why you get the flare. And the flare is usually, I don't know, like this, not symmetrical, flaring at different locations, whatever, flaring at different rates too.
Is there any cloudiness to this tube at all, or does that only occur when the bacterium are completely mobile and all throughout the tube?
It's cloudy in the flare, okay?
Okay, that makes sense.
But the flare will look closer to, let me see right here. There we go. It'll be closer to that, very, very cloudy in the flare. It may be a little lighter than that, but it'll be darker than that because this is where the bacteria everywhere. The bacteria there are only on the stab. And when it flares, it'll be closer to that. Probably a little lighter than that, but darker than that. That was a good question. I've never had that question asked. All right, let me shrink that down. All right, let's talk about the hydrogen sulfide production test. When the bacteria make hydrogen sulfide with three different media, you can test for the production of hydrogen sulfide. They each have a reagent in them that will form a black precipitate with hydrogen sulfide. So it doesn't matter if you grow it on TSI agar, Sims media, or hectone enteric agar. If hydrogen sulfide is made, you'll always see blackening of the media. We have one right here. That is a Sims tube. And usually you find the media turning black in the lower portion of the term, not in the top portion of the term. And the reason is the hydrogen sulfide typically is made anaerobically. And so at the very top of the tube, even if you have bacteria up there, you do not have hydrogen sulfide generally made. It's in the lower portions of the tube.
Are you looking for the indole production up at the top, or is that with just a completely different test?
In the Sims tube, you're actually running three tests at one time. And the indole is one of the tests you're running. It will show only in the top, but you would find it within the Sims tube. In this tube, I'm not sure they even added Kovacs reagent because I don't see a solution on top of the liquid here. So they may not have added Kovacs reagent yet to this Sims tube because they're just trying to show you the hydrogen sulfide production.
Okay.
And
Is
you
that
can,
Oh,
what?
I'm so sorry. Is that something we should just kind of in the back of our mind keep in mind just in the event if, say, this was a positive, sulfide production is clearly there, but if, say, it was red or pink on that upper top layer for a positive indole production as well, should we
Yeah,
look
I'll
out for
talk about the Sims test in a little bit, in which case I'll show you where you're looking at the three different tests at one time in one tube. Okay.
that? Okay.
But for here, I'm only trying to show you hydrogen sulfide production. And let me go here. I'll show you Sims tube. And that right there, I'll come back to it. There's another Sims tube. And there you can see black from hydrogen sulfide production. And there's a reddish color that's from indole. And the motility I'll talk about later. Let's go back to the TSI tube. There we're seeing black once again, only in the bottom portion, because hydrogen sulfide, like I said, is usually made anaerobically. It is not made aerobically. And that's why we're not seeing it black up here. And then on hectone enteric agar, you can also test for hydrogen sulfide production. And if you have hydrogen sulfide being produced in the colonies on ectone and enteric agar, the colonies will be black. If there's no hydrogen sulfide being produced, the colonies will not be black. They'll either be their normal color, or if they produce an enzyme which digests the sugar lactose, they'll be salmon colored. Salmon colored is yellow-orange. And we'll talk about this test later, okay? I just wanted to show you hydrogen sulfide production. Doesn't matter which test you use to get it. If it tests positive for hydrogen sulfide production, the tube will turn black. Any questions about any of that? All right, 3 organisms which turn the tube black or produce hydrogen sulfide are Citrobacter, Salmonella, and Proteus species. Next, let's talk about the triple sugar iron agar slant, or TSI slant, or TSI test, whatever you want to call it. This test has 3 different sugars in it: sucrose, lactose, and glucose. But note, the glucose is 1/10 the concentration of the lactose and the sucrose. The media also has proteins in it. We'll talk about that in a little bit. And then phenol red as the pH indicator. And then sodium thiosulfate and ferrous sulfate to detect the presence of H2S production. They are what turn the tube black if hydrogen sulfide is made in the TSI tube. How you inoculate the TSI slant is first you get a needle, and then you come up to your bacteria. I don't know if you can see this, I'm trying to show you. And then you get bacteria on the tip of the needle. And then you inoculate the needle into the agar. You should come down further than this because that's only about halfway. You should come down about 2/3 of the way to the bottom from the lowest part of the slant. Why you want to come down 2/3 of the way is because you want the bacteria to be growing anaerobically in the butt of the slant. And here, the growth, even though it's in the agar, that's so far close to the air that that's probably growing aerobically. So right here, the only portion we have, which is really anaerobic, is the very bottom where the slant is. And that's why you should take the needle about 2/3 the way down the tube from the bottom of the slant. So go a little more than halfway. You do not want to go all the way because the needle is very fragile and very easy to bend, and you don't want to bend it. I'll tell you why in a little bit, but you don't want to bend it. And if you go all the way down, you are likely to touch the glass, and that can bend the needle. So you only want to go about 2/3 of the way down the agar, 2/3 from here. Any question about that? Anyways, that's to get growth of the bacteria growing anaerobically. You then pull the needle straight out and take your needle once again and go through the bacteria colony, getting bacteria on the tip of the needle. And then you inoculate the slant with the bacteria, just moving in an S-shaped motion. Do not cut into the bacteria. You just want to use it like, I guess you're painting the slant with the bacteria. And for the slant, you don't have to use a needle. You could use a loop. The good thing with the loop is that you're less likely to cut into the agar. If you cut into the agar, you're likely to remove all the bacteria from the needle, and then the upper region won't have any growth, and all the lower region will have growth wherever the you cut into the agar. So you don't want to cut into the agar. You do not want to put the loop into a TSI tube, inoculate the butt, because that will damage the loop. The loop is fairly fragile. This is 1% agar, and you can break the loop by sticking it in 1% agar. Doesn't mean you will. I'm saying you may break the loop. And then that would be expensive for the lab. Any question about any of that? Anyways, that's how you inoculate the tube. You then incubate it for 48 hours. And then you pull the tube out and you look at the growth in the butt and the growth on the slant, looking for a large number of different tests. You're looking at the production of hydrogen sulfide, where the tube will turn black. You're looking at the fermentation of the sugars, where the tube will turn yellow. If you have glucose and one other sugar fermented, the tube will be yellow on the slant and yellow in the butt. If the tube,
Is it possible for any of the tube to be red in number 4 for any reason?
no, because the sugar is being fermented in both the butt and in the slat, and so it should turn yellow from the production of acids. Anyways, you get a tube that has yellow in the butt and the slant, tube 4, then you know at least glucose is fermented, plus at least one other sugar, either lactose or sucrose, or both of them are fermented. Gas is also produced in tube 4, and that's why there's cracking of the media shown here, and then the bubble shown here. If you only have cracking, gas is produced. If you only have a bubble and there's no cracking of the media, gas is also produced. Any question about tube number 4? All right, let's talk about tube number 3. If you have the butt yellow and the slant reddish or pink, what happened was only glucose was fermented. And glucose is at 1/10 the concentration of sucrose or lactose. And so all of the glucose in the slant got used up. And rather than die, the bacteria then start utilizing protein. And if you utilize protein, you'll make urea, which is basic. And that will change the color of the slant from yellow to pink from the basic products. So if we were to look at tube 3 earlier, like after 4 hours or after 8 hours of growth, the slant would have been yellow. But after the time of the tube, which is 18 hours to maybe 24 hours, the all the glucose got used up in the slant, and then the protein was used, making the basic products, and the color reverted from yellow to pink. And pink is just basic color. If you have no color change, meaning red, the pH stayed around neutral. So that's either neutral or pinkish. Now the bottom of the tube will remain yellow because in the bottom of the tube, glucose is fermented slower than on the slant, which is growing aerobically, and the bottom of the tube is growing anaerobically. And so the glucose will be used up slower. And in the time frame of this tube, the bottom should remain yellow because not all the glucose will be used up because it's being fermented slower. So it won't start changing red, but it will be yellow because the glucose is being fermented, creating the acid products. Any question about 3? Obviously, it's not black, so there's no hydrogen sulfide being produced. There's no bubble. There's no cracking of the media, so there's no gas being made in the fermentation of glucose. Any question about tube number 3? If not, let's go on to tube number 2. Tube number 2, of course, has black, so hydrogen sulfide was produced, and it's only in the bottom of the tube, meaning hydrogen sulfide is normally made anaerobically, and that's why it's not being made on the slant. It's aerobic growth. The slant is pink, but the bottom is yellow. Now you can't see the yellow, but wherever there's black, you can assume that the sugar is fermented because hydrogen sulfide production is made only when sugar is fermented. And so if the tube is all black, then we know it is yellow until it became black. And so once again, it has yellow on the bottom, pink on the slant. So only the sugar glucose is being fermented, and all the glucose got used up on the slant, and that's why the color changed to pink. Also in tube 2, there's no gas being produced because there's no breakage or, or disruption of the agar. It's all intact, and there's no gas bubble. So no gas is being produced. In tube 1, we have no sugar being fermented, no gas being produced, and no hydrogen sulfide being produced. And so that tube remains its original color, which is usually red. It looks like it's starting to become a little bit basic there. And that's probably because of CO2 from the air joining with the media, forming. No, that wouldn't be right. That would be acidic. Never mind. It's not from CO2. It could be the bacteria using the protein in the media, making urea, because it does look a little pinkish right there. Any question about TSI? It is a complex test looking at many different things going on at once. And the best thing for me to say is just know this table.
Um, I do have a quick question.
Yeah.
Um, when you're asked just solely whether a TSI tube is positive or negative, you're not looking specifically for glucose, lactose, or H2S, just TSI. What are you looking for to determine whether that is a positive or a negative TSI test?
Okay, for the TSI test, you're running multiple things at one time, and so you don't say the test is positive or negative. For tube 1, for example, we have either K over K, or you could write negative over negative, meaning negative in the slant, negative in the butt. For tube 2, I guess I never mentioned that. This is K or negative in the slant, and then A or acid in the butt. So it should be yellow, and that's why we're going A. For tube 3, we have negative in the slant, and then A in the butt. Oh, wait a minute, go back to tube 2. We also have the mention that hydrogen sulfide is being produced there. For tube 4, we have A in the slant over A in the butt. And so we say it is A/A. And then we also add that gas is produced for tube 4. So that's how you score the TSI test. You don't ever say positive or negative unless you're doing something where you say hydrogen sulfide being produced, and you're only looking at hydrogen sulfide. So instead of saying on the table TSI, you would say H2S, maybe production, or just H2S, and that would be H2S positive. All of these would be H2S negative, meaning the tube 1, tube 3, and tube 4. Any question about that? All right, let's move on to the next test, and that is the sulfide indole motility test, or the SEMS test. And SEMS stands for S for sulfide production, I for indole production, M for the motility test. So you're running 3 tests at one time in the SEMS test. What you do is you stab inoculate the SEM media with a needle, and you um, stab it down, straight down, and then you pull it up, straight up, so that the tube only has one single stab mark, meaning you don't put it in something like this and then pull it out something like that, so that it has only one stab in it, meaning you don't have a smear in it or a slice in it. And then you incubate it, being very gentle so you don't disturb the stab mark, and incubate it for 24 or 48 hours, something like that. And then you add Kovacs reagent to the tube, looking for indole production. And if the tube turns pinkish or purplish in the Kovacs reagent, then indole was produced. And so this is indole positive. That's indole negative, where it changes color to yellowish, and that's sort of a very light yellow or off-white color. That's also negative for indole production. You then look at the tube to see if the lower portion of the tube is black, and that's hydrogen sulfide production. So tube 1 is positive for hydrogen sulfide production. The control, tube 2, tube 3, tube 4, and tube 5 are all negative for hydrogen sulfide production. You then look at the motility in the tube. And if the bacteria are only growing in the stab mark, like tube 2, tube 3, and tube 5, I don't know if you can see that. Let me blow that up. There you can see the bacteria in the stab. That one's really hard to see, but there's the stab there. And then this one is better. Bacteria is only growing in the stab. So these bacteria in tube 5, tube 3, tube 2, and tube 1 are not motile. Tube 1 may not even have bacteria in it. I don't know. And then tube 4 is a good example of flaring, meaning we don't have the bacteria growing everywhere in tube 4, but we do have it growing here and here. So this would be a very large flare. And then we have it growing in the stab mark. So this is slightly motile bacteria. It's not going everywhere, but it is motile. And that you call motile positive. You could say slightly motile positive if you wanted. And then in tube 1, you'll notice that it's black everywhere. That's because the bacteria have swam out of the stab mark and then went everywhere. And if you could see the bacteria, it would be like this everywhere, meaning cloudy everywhere. And that's why it's black everywhere. But in this case, the bacteria are also producing hydrogen sulfide, and that's why it's turning black everywhere, except maybe up there. And it doesn't turn black where the bacteria are growing aerobically. Because hydrogen sulfide is made usually anaerobically. Anyways, that's the SEMS test. Any question about the SEMS test? All right, let's take a look at growth on a McConkey's auger. McConkey's is testing two things at one time. You're testing to see if the bacteria are gram-negative or gram-positive, because the chemicals in McConkeys keep most gram-positives from growing, but most gram-negatives can grow on McConkeys. So if there's growth, then it's likely to be gram-negative. We're also looking at the fermentation of the sugar lactose in McConkeys. If the bacteria cannot ferment the sugar lactose, they will be their normal color. The media will be its normal color. And in this case, the bacteria are either white or off-white. If, on the other hand, the bacteria can ferment the sugar lactose, they will turn pink in their colonies or their streak, and then the media around the growth will also turn pink. And the reason for the pink color is the sugar lactose, when it is fermented, will make acid byproducts, and the pH indicator will turn pink if the media is acidic. So a lactose fermenter, a non-lactose fermenter on McConkeys, and both of these species are gram-negative because they're growing on McConkeys. Any question about any of that? All right, let's talk about growth of the bacteria on an HE or hectone enteric agar plate. We have three things we can look at in the HE plate. You can first look to see if the bacteria produce hydrogen sulfide. If they do produce hydrogen sulfide, the bacteria will be black in color. If they do not produce hydrogen sulfide, they will not be black in color. They'll either be their normal color, which in this case is white or off-white, but it could be whatever the bacteria color is. If the bacteria form yellow colonies, then they'll be yellow if they don't ferment the sugar, if they don't make hydrogen sulfide production, and they don't ferment the sugar lactose. They'll be their normal color. Any question about any of that? All right, let's see, the first thing you're looking at is, do the bacteria grow on the HE plate? If they grow on the HE plate, then they're likely to be gram negatives because they can grow on an HE plate. So all three of these species are gram negatives because they're growing on the HE plate. Gram positives are selected against, and so they usually do not grow on an HE plate. So that's the first thing we're looking at. The second thing we're looking at is hydrogen sulfide production. And then the third thing we're looking at is the fermentation of the sugar lactose. If the bacteria cannot ferment the sugar lactose and do not produce hydrogen sulfide, they will be their normal color, which in this case is white or off-white. If they do ferment the sugar lactose, when grown on an HE plate, they will be salmon colored. Salmon color is a yellow-orange color, and that's from the pH being changed to acidic by the fermentation of the sugar lactose. If, on the other hand, the bacteria produce hydrogen sulfide gas, all you'll see is black colonies, and you cannot tell if the sugar lactose has been fermented or not fermented because the colony is black. So we don't know what color the bacteria would be if hydrogen sulfide was not produced. Any question about HE? All right, Salmonella is a bacteria which will be black on an HE plate. E. coli, when grown on an E. coli McConkey's plate, will be pink in color because it can ferment the sugar lactose. All right, any questions about the lab module? If not, let's go to the worksheet. I think it starts now. Nope. Let me talk about the lab module a little further. All right, so you're not going to be physically doing any of these activities in this lab because everything we do will be done online. However, you will be held responsible for knowing how to perform and interpret the following tests. We have a little video on the carbohydrate fermentation test, so watch that video. We have another little video on the SEMS test, where 3 tests are being run at one time, so watch that video. We have a little video on the MRVP test. Remember, with this test, you start this test by inoculating 1 tube with your test organism, incubating it, and then after incubation, you split that tube into 2, 1/2 the tube being used for the MR test, the other half for the VP test. And then watch a little video on the TSI test. And then we have a little video on growing bacteria on the McConkey's agar. The microbuzz no longer works, or you must pay to access their website, but you should be able to get all of the information that are required for this lab from this lab module, with the exception of the very first question in the table. And then we have some case studies for you to work on in the in this lab as well. So let's go to the worksheet now and go through the questions. So question 1 is fill in the table, reviewing how to interpret the following test by filling in the table. You just need a description. The first cell of the test has been, or table has been filled in for you. For oxidase, the cells or the spot turns blue. You need to say what a negative result will be for the oxidase test. This is the only test that is not discussed in this lab, and you can get the oxidase test in a previous lab, or you can get it online either way. For the indole test, tell me what the positive result is and the negative result. For the MR test, tell me what the positive result is and the negative result. For the VP test, tell me what the negative result is, the negative result is, and tell me what the positive result is. And do the same for the citrate test. Tell me the positive result and the negative result. Then move down to the carbohydrate fermentation and tell me what the positive result will be if the sugar is fermented. So tell me what the color is, for example, if the sugar glucose was fermented. And then tell me what the color of the tube will be for the negative result. And then for carbohydrate fermentation, tell me what the positive result will be for gas being produced and the negative result for gas production. So you don't say AG here for positive result because we're not talking about gas being produced here. Okay, we're only talking about gas being produced here, and this is a little odd, and that is we're talking about gas being produced. We're not talking about the sugar being fermented, but to get gas production, you have to have the sugar fermented. But you only need to talk about gas production here. If you need more space, just make the cell larger. You can also write here or write up here or down below the table. All right, let's move on to the triple sugar iron agar slant. Tell me what the positive result will be if glucose is the only sugar that is fermented. And for all of these TSI, at least glucose, lactose, you need to state the color of the slant over the color of the bud. Or you can use the abbreviation, something like A for the slant over A for the bud. And then give me the negative result. Once again, you should have the slant over the bud. And then tell me what happens in the positive result if lactose and or sucrose is fermented in addition to glucose. Tell me what the positive result will be, and you need to state it in terms of the slant over the bud. And then tell me the negative result of lactose and or sucrose being fermented. Then tell me the positive result of hydrogen sulfide production, and then the negative result. And then tell me in the TSI agar slant the results of a positive result for gas production, and then a negative result. Remember, there are two possibilities for being positive. You really should either find a term for both or you write a description in here. Move on to the SEM auger test. SEM stands for sulfide indole motility. So tell me the positive result of the sulfide production test and the negative result. Tell me the positive result of the indole production test and the negative test result. Then tell me the positive result if your bacteria are motile, and tell me the negative result if your bacteria were not motile. And then lastly, give me the color for MAC and HE if lactose is fermented in the positive result, and if lactose is not fermented in the negative result. Any questions about this table? All right, for 2A, name the reagent that must be added to detect the presence of indole. If indole is present, the test will show what color. So put that for A2. That's A1. All right, methyl red is the pH indicator that remains what color under acidic conditions. So name a color for B1. And then methyl red is a pH indicator is used in what test? So name one test using methyl red indicator. For C1, which test uses Barrett's reagent A and Barrett's reagent B to detect the presence of acetone, a neutral product formed during glucose metabolism? If acetone is present, the test will show what color. So fill that in for C2. The Simmons citrate test uses the pH indicator. So name the pH indicator right there. And this pH indicator turns blue color under either acidic or basic conditions. So you need to either write acidic or basic in the blank there, or you can highlight acidic or basic or underline acidic or basic. Just make it clear to me what your answer is. For F, bacteria that possess this cell structure test positive for motility. So tell me what cell structure bacteria will have if they are motile. 4, G. 3 organisms that produce hydrogen sulfide are 1, 2, and 3. So list 3 organisms that produce hydrogen sulfide. That was mentioned in the lab module. You can also find it online. If you don't have the same answer as the lab module, it's not wrong as long as your answer is correct. H, if an organism ferments glucose but not lactose or sucrose, the TSI will be what in the slant and will be in the deep will be what or the but. So for H1, tell me the color of the TSI slant. Organism ferments glucose. And then tell me the color of the slant in the butt if the organism ferments the sugar glucose. J, MAC, and HE plates are both what? In that they inhibit the growth of, and then state either gram positive or gram negative. So MAC plate for J2 state either gram positive or gram negative, or you can highlight and underline. And then MAC and HE are also what? In that they separate various bacteria based on the visual differences from sugar fermentation. All right, any questions about the worksheet so far? Not. Let's talk about the case studies. We have a table showing the results of certain tests for the bacteria in the case studies. Only use this table for the case studies in this lab. Do not use it for the table in your lab report, because not all the species are being used in this lab, as well as this is meant to be the lab, not the unknown project. Anyways, use this table to identify the case studies, and then use this table for when you're going through the case studies. Put in the oxidase result for case study 1, put in whether case study 1 ferments the sugar glucose, ferments lactose, etc. Put in the results of the YP or that's the results of the TSI tube. I don't know what I was saying earlier. Um, and then put in the results of the hydrogen sulfide, the results for motility, indole, MR, VP, and citrate. Put that in and fill in this table, what the results are, and then use this table with the table above to identify the bacteria. For example, if you have an organism which is only fermenting a sugar, then you know you have a member of Enterobacteriaceae. I shouldn't say it's fermenting the sugar glucose. Let's put it that way. Actually, it's all right if it's fermenting any sugar, because only the family Enterobacteriaceae can ferment a sugar. Pseudomonas aeruginosa or Pseudomonaceae, uh, none of them can ferment any sugars. And then write your answer for the species causing the infection in the case study number 1, 2, 3, 4, and 5. The case studies are in the lab module. I thought I switched that. So I'm only going to read one of these. I'm going to take case study 2. Oops. Going through the example. Mikey talks his mom into buying a pet map turtle. He does well at changing its water and probably feeding the turtle, but he's not necessarily diligent about washing his hands afterwards. About 2 weeks later, Mikey develops diarrhea, nausea, and fever. The mother takes them into the pediatrician, and the pediatrician orders a fecal specimen for routine culture and sensitivity testing. Here we have some bacteria growing on McConkey's media, and the bacteria of interest are pointed in the arrow shown here, and that tells you something that is growing on a gram-negative plate, and the bacteria of interest are not pink. So you can look up what the results of that means and fill it in the table above. Here is the bacteria growing on an HE agar plate, and there you can see the bacteria are largely black. Further tests were run. We have the SIMS test here looking at sulfide production, indole production, and motility test. So write your results there. This is just the room. And then, oh, wait a minute. I thought that was a chair. I wasn't looking very good. Um, this is the MR test, and there's the MR results. This is the VP test. That's the VP result. This is the Simmons citrate test, and there's the result of the Simmons citrate test. And then this is the result of the TSI tube. You can see several things going on there. Then use the table for identifying the bacteria. Make sure you don't use this table for your unknown project because you'll be wrong, because this is only looking at, you'll most likely be wrong. This bacteria, this uh, chart and this whole lab are only looking at two families of gram negatives. And then let's come back to here and then state in case study number 2 what the bacteria is which is causing the infection. And then do that for case study 1, case study 3, case study 4, and case study 5. Any questions about any of this lab?
So if we're going through this and for some reason what we're interpreting the test results as, if they do not match any organisms on the table you've given us, we are incorrect, right?
I believe that's so because this lab is talking about certain bacteria, and it will be one of the bacteria in the table.
Okay. So if we're interpreting things wrong, then that's because we're wrong and not the table. So, okay. Thank you.
To my knowledge, there's no mistakes in this lab, such as in the table. Why do you ask that question? Did you get something that didn't fit the table?
I think I'm just having probably a couple of hiccups really reading a TSI slant with the glucose and the lactose versus the K and A or
Uh-huh.
any just kind of those readings.
Uh-huh.
So I was just curious. So clearly it's I'm reading the tests
Yes, TSI
incorrectly.
could be being read incorrectly, and that test has something like 4 different things going on, and so
Right.
it can be confusing.
Okay.
Hopefully, you got the hydrogen sulfide production correct.
Yeah, that's the easy
It's the
one. But
easy part, yeah. All right.
sometimes differentiating between the glucose fermentation and then the lactose sucrose and what that would look like
Mhm.
if they were both fermented or only partially is a little gray area. So,
Okay. Well, if there's no hydrogen sulfide being produced, then the bottom of the tube will be yellow and the top of the tube will be red if only glucose is fermented. If glucose is fermented and one other sugar, or maybe both of the other sugars, are fermented, then both the slant and the bottom will be yellow.
okay. Thank you.
All right. All right. I will be here until 8 o'clock. We'll go 8:15 to answer any questions. If you have any questions, just use me, and I will be here until the last student leaves or until 8:15. All right, get started on the lab. Okay, there was a question about the table. If the table doesn't have a plus or a minus in it, treat it as a variable, meaning if it doesn't have a plus or a minus in it, what's an example? Treat it as a variable, meaning that it could be either plus or minus. Must be in the worksheet. For example, in the table, it has Citrobacter furundi. It has indole. It doesn't have a plus or a minus in it. And so treat that blank as either a plus or a minus, meaning if Citrobacter furundi, you're running that one and you have the results for indole, whatever the results will be, it will fit Citrobacter furundi.